Hello, this is Dr. Carmel Rogers with 3 questions asked, and today we're going over DNA and RNA. Some of the learning objectives here would be to understand the base pairing rule. We'll also be able to identify complementary and identical nucleic acid strands, and we'll practice with 3 questions so that way you can apply the information from understanding more about nucleic acids. This is our first question. Write the complementary DNA sequence to this sequence. Let's go ahead and start by a bit of a review. And we're going to talk about the central dogma. The central dogma of molecular biology goes over or explains how information is transmitted through the cell. And so we start off with this cell. It's a eukaryotic cell. It has a nucleus, and within the nucleus, we have our DNA. And notice how DNA is double-stranded. Going from DNA to RNA is transcription. The enzyme that's responsible for transcription is RNA polymerase. So that's going to use DNA to make a copy of RNA. And then we have RNA to protein. And that process is translation. Now, the way to differentiate, or one way to remember the difference between transcription and translation, is that transcription has a C right here, whereas translation has an L, and so C comes before L. So going from DNA to RNA is transcription, so that's the earlier process, and then RNA to protein is translation. Now, going from RNA to protein, we need ribosomes. It almost looks like a hamburger. You have a large subunit at the top here, and then a small subunit, and then the mRNA fits right in between those subunit buns. Okay, so a little bit about DNA. I want to take a closer look at some differences here, and we'll also get into polarity of the strands. Now, with DNA, we know that there are bases, and the bases here are adenine, thymine, guanine, and cytosine. The bases are going to be at the middle of the DNA ladder, whereas the ribbons on the outside would represent the phosphate and sugar backbone. Here it goes over the 3' and 5' nature of the strands, and this is called anti-parallel. If we look at part C, it's also showing 5' on the left at the top, and it has an arrow that goes to 3'. What that represents, at each of the nucleotides, we have a base. This one is for adenine in red. We have the sugar in blue, and then we have our phosphate group. That's what makes up the nucleotides here. The phosphate is at the 5' end, right here. So here's our 5', and that's where we see the phosphate. So at the 3' end, we see the hydroxyl. Okay, so that is what is referred to as polarity. And with DNA, one strand will go from 5' to 3', and the other strand is anti-parallel. And so notice how we have 3' and 5' on the opposite strand. A will pair with T, and we can see that at the top. So adenine going to thymine makes 2 hydrogen bonds. Then we have C with G, and we get 3 hydrogen bonds in that base pair. So the hydrogen bonds are important because they help to stabilize the interaction of the base pairs. Now, since we've talked a little bit about the DNA structure and the polarity, now we can go a little bit into RNA. So RNA has adenine, cytosine, guanine. And instead of thymine, it will have uracil. So this is one of the big differences, okay? Uracil, if we were to pair that with DNA, uracil will pair with an A. We still get the regular C with G, and so uracil with A. And these are some of the comparisons that you want to keep in mind. Now, we have messenger RNA, and we also have the transfer RNA. As we work through some of our questions here, it will be more clear how you're going to do the base pairing. I want to give you a quick acronym for how to solve problems. And with the acronym, we're going to use CTM. These are different types of strands that you'll encounter when doing nucleic acid comparisons. The first one is coding. Next one is T for template. M for mRNA. And then that last T for tRNA. And with the acronym, you can just think of catchy with the C, so that relates to coding. Trendy starts with the T, just like template. Then we have music relating to the mRNA, and then time, and that T relates to the tRNA. So we just want to remember the order of these strands: coding, template, mRNA, then tRNA. Catchy, trendy, music, time. Just remember those first letters there. The way that we'll use this is, I have an example, and we'll do 5' A, C, T, G, C, A, 3'. So that's going to be our coding. Next, we have template. Now, comparing coding with template, we are going to have anti-parallel strands. So If the coding starts with 5 prime on the left, the template will be 3 prime on the left. Now we use our base pairing rule. Now both coding and template are DNA. So we're going to use A, C, G, and T bases. A pairs with T, G with C, T with A, and this G with the C with the G, and the A with the T. And we have 3 prime on the right for our coding, so that would be 5 prime on the right for the template. All right. Now let's find the messenger RNA and the tRNA. If we use the template strand and go to the messenger RNA, those would be complementary. We have 5 prime. With the messenger RNA, remember it is a type of RNA, so it's going to have uracil and not thymine. We still have the base pair for T, which is A. Then for G is C. And then for the A, instead of T, we're going to put U for uracil. Okay. Go ahead and try the last 3 bases. Feel free to pause your video, or if you need to rewind, you can definitely do that too. So I'll give you a few seconds, see if you can figure out what the last 3 are, and then we'll do it together. I hope you're enjoying the session. Please let me know below in the comments other topics that you'd like for me to cover. And also, like, share, and subscribe. That will help me to grow my channel. Thank you for watching. All right, so we're looking at the template, and for the mRNA, that's going to be a G. Then we have a C, then we have an A, and they're anti-parallel at the end, 3 prime. Now we're doing the tRNA, which is complementary to the mRNA. So again, we have to make it anti-parallel. So the tRNA will start with 3 prime. Now, tRNA is a type of RNA. Instead of thymine, we have uracil. Then we have our guanine, adenine, cytosine, guanine. And we're going to end up with our uracil and then 5 prime. How did everyone do on that? Let me know in the comments if you were able to get all 4 strands. I want to talk a little bit about using the acronym. So catchy, trendy, music time, coding template, mRNA, tRNA. If we look at the strands, notice that every time we go down one level, they are complementary. Coding to template is complementary. Template to mRNA is complementary. And then the mRNA to tRNA is complementary. Now, if you were to go down 2 levels, so going from the coding to the mRNA, That is identical. And what do I mean by identical? Notice coding starts with 5' on the left, just like the mRNA with 5' on the left. And they both have A, they both have C. Remember, mRNA is a type of RNA, so instead of thymine, it will be uracil. They both have guanine and then cytosine, followed by adenine, and then we end up with our 3' N. These are identical, coding and mRNA. Now let's have a look at the template, and what we'll see is that comparing template with tRNA, we also get identical strands. So the template with tRNA would be identical. And let's have a look. So they both have 3' on the left. We have a T in the template, which is for DNA. And the tRNA is a type of RNA. So instead of T, we get uracil. They both have G, then A. Then we have our C and G. And again, DNA has the thymine, whereas the RNA is going to have the uracil, and it ends up with the 5'. Okay, feel free to rewind if you need to go back over this. Now we have a good understanding of how the bases will pair together, and we can solve our first question. And it says to write the complementary DNA sequence to this sequence. If you need to use the acronym, feel free to do that. With this one, it's pretty straightforward. They don't give you whether it's coding template, mRNA, or tRNA, but the hints here are that it's complementary and that it's DNA and not RNA. Okay, so we know it's not going to have any U's because it's DNA. Now we're going to follow the anti-parallel rule. If the complementary strand has 5', its complement is going to have 3'. The complement for G is C, for A is T, for T is A. And then we have the complement for this C is a G. The next one is also a C at the top, and so that will pair with the G. Then we have an A, then T, A, T, C, T, C. And if the top has 3', then the bottom one will have 5'. How did you do with that one? Was it pretty straightforward? Let's go to the next one. And in this next one, we actually get some options to choose from. I will show this to you for a few moments and go ahead, make sure you have your scratch paper. You can use our acronym, catchy, trendy music time. Okay, I'll give you a few moments to solve this one. Let's work through it together. So, what mRNA sequence is generated from this template strand? Now, if we look at our acronym, template is this T right here, and it's a type of DNA strand. And then we have M for the mRNA, and it is RNA. And that's what we're looking for. They give us template, and we're solving for RNA, mRNA. So we can put a little question mark by our mRNA because that's the one we are solving for. Now, template compared to mRNA would be complementary. Remember, every time we go down one level, it's complementary. If we go down two levels, it would be identical. So we can go ahead and do our pairing. So on the left, we have 3 prime, then C. And if the template has A, then the mRNA will have U. If the template has T, the mRNA has A. Then we have G, G, A, U, A, U, C, U, C, and 5 prime. Now let's look at our answer choices. We know that we're solving for the mRNA, and mRNA doesn't have any T's. So we can automatically get rid of the bottom one because that has some T's in it, okay? Now, the other answers choices, they all have 5 prime on the left. But the way that we solved it, we have 3 prime on the left. Now, the point I want to get across is that every sequence is equal to itself when you write it backwards, but it has to be consecutively backwards. And so this is how it will look. We're just going to start at the 5 prime end and then work our way to the 3 prime end. We have 5 prime CUC, and then UAU, and then AGG, and then AUC. And then 3 prime. Okay. So that's just one thing to remember. If you don't see the answer in the way that you solved for it, just write it backwards and see if you can align it one of choices. So we have, let's take a look. So the first one has 5 prime GAU, that's not right. The second one has 5 prime CUA, that's not right. The third one has 5 prime CUC. Then we have our UAU, then AGG, and then AUC, and then 3 prime. So C is the correct answer. Feel free to pause or rewind to make sure that you understand how we got C. Let's move on to our last question. What is the coding sequence for this mRNA sequence. All right, so we can go ahead and write down our acronym, coding, template, mRNA, tRNA, catchy, trendy, music, time. That's the order that you want to put it. Every time we go down one step, it is complementary. And if we skip a step, so going from coding to mRNA or going from template to tRNA, that would be identical. Okay, so we want to remember this. Now we're doing coding and comparing it to mRNA. Notice we have coding at the top and it skips a step, and then we have our mRNA. So this is an identical comparison. Got that? If they're identical, we just have to remember that RNA has uracil, whereas coding is a type of DNA, so it's going to have thymine. And the only thing we really need to do is change the U's to T's. So we're solving for the coding DNA. We can put a little question mark because that's the one that we're solving for. And they give us the messenger RNA. The polarity will be the same. We'll have 3 prime. The C's are the same. The G's are the same. Instead of U, we'll have a T. The A's are the same. Instead of U, we have a T. The C's are the same. Then we have G, C, A, T, A, G, 5 prime. Okay? Now let's align this with our answer choices. And we can also remember what I had mentioned earlier. If you don't see your answer in the way that you solved for it, you can also write it backwards. So let's have a look. Sometimes they may present it exactly in the same way you solved it, but if not, then we know what step to take. The first one starts off with 5 prime GCA. Okay. If we look at our 5 prime end, we have 5 prime GAT. So this is incorrect. In the second option, we have 3 prime C, G, and U. Now remember, we're solving for DNA, so DNA is not going to have uracil. So therefore, we can get rid of B also. In the third option, it has 5 prime, then GAT. That looks really good. So in the one that we solved, we have our 5 prime end. So a 5 next to a G, next to an A, next to a T. Then we have, and we're just reading it backwards, then ACG. So that would be the ACG. And then CTA. We have our CTA. And then TGC 3 prime. TGC 3 prime. We found it. And just to prove that these two are the same sequences, we can just go ahead and write it backwards. And the way that it looks, it would be 3 prime So 3 next to a C, next to a G, next to a T. Then A next to a T, next to a C. Then G, C, A. Then T, A, G, and 5 prime. All right. And these two are the same. Just to be thorough, let's have a look at D. D, we have 5 prime next to CGT. And in ours, we have 5 prime next to GAT. So that's incorrect. All right, so that is 3 questions solved. I hope you learned something new. What other topics are you interested in? Please let me know below in the comments. I look forward to working with you. I do one-on-one sessions and also group sessions. And let's come together and get closer to your educational goals. Bye.